It's great to be here. So yeah, as was just mentioned, I've always been fascinated by collective behavior. Ever since I saw footage like this of schooling fish on a coral reef or flocking birds, I wondered both how and why organisms like this, who happen to be unrelated to each other, coordinate their behavior in such a fantastic way. And it's really a long-lasting challenge because we still only have a very rudimentary understanding of both why and how we see these types of behaviors. So, as was mentioned, we study a very wide range of organisms within our department. We literally study the simplest organism on the planet. This is Placozoa. It's a collective of cells. It's in the base of the metazoan tree, arguably the simplest metazoa in life. And of course, the reason we're interested in it is if you track the nuclei, you look at their density, or look at their motion, the direction that they're traveling, you can see these waves that are indicative of local interactions leading to long-range patterns within these types of systems. As we sort of go to more advanced organisms, again, we find it's relatively local interactions among organisms such as these locusts. These are a devastating pest that impact the livelihood of 1 in 10 people on the planet through food shortage, but hardly anyone studies because the people it impacts are subsistence farmers. Yet, these types of organisms, when you look at this behavior, you're often tempted to think, well, they're cooperating. They're doing this together for a common good. But way back in sort of the early 2000s, I developed software that allowed us to track the motion of these individuals. Now, at the time, the software was the most advanced software out there, but you can see it's relatively primitive when you compare it to the stuff you've seen earlier today. But this did allow us to reveal that in actual fact, these swarms are driven by cannibalism. They run very short of essential nutrients, and what's better than a perfectly packaged locust for all of the nutrients you need? And so every individual is trying to eat those ahead and trying to avoid being eaten by those behind. The outcome is collective motion, which also allows them to leave the nutrient-poor areas and find food.
Ian, that's like the bumps, no? In Oxford and Cambridge, the boat race.
Yeah.
It's just the same
Everyone's
thing.
chasing. Yeah. And the really exciting thing about studying collective behavior, both in the lab and the field, is the advent of deep learning. And this is a modification of the LEAP software from Josh Shavitz in Princeton. This is sped up, but it allows us to track the full body posture of thousands of individuals simultaneously, plus we have little barcodes on the individuals to allow us to identify who is whom. But we also study animals in their natural environments as well as the field, including locusts. These are arguably the most complex organisms we study. These are human beings, and this is in a railway station in London. And unbeknown to these pedestrians, we actually have actors within the crowd subtly manipulating subconscious aspects of attention. In this particular study, this was prior to the London Olympics, and they were very worried about crowd security. We had the idea that people are well known to be able to detect aberrant behavior, but there's a phenomenon called bystander apathy, where people don't actually do anything about it. But if we can detect people detecting suspicious activity, and we can show that there's social contagion in the crowd, which there is, then perhaps we can use this to better understand and predict where outbreaks of violence, for example, may happen. And so a hallmark of collective behavior in the natural world is that there's information transfer, whether you're dealing with a collection of neurons or dealing with a collection of organisms. And so when you look at footage like this is a trumpetfish attacking silversides on a coral reef, you can almost imagine why in the 1950s and 1960s, people believed there must be thought transference among the individuals to give rise to the high synchrony that you see. But what we now know is that, and as I'll describe to you shortly, relatively local rules applied by each individual within the group can actually explain many of the patterns that we see in nature. And so while many people study the individual mind, the individual brain, what I've become fascinated about is how social connectivity, and this can arise in a variety of ways, connects minds together. Can we think of collective minds or collective cognition within animal groups? And if so, what does that look like? And how does that differ from the types of cognition that we're used to thinking about within the individual brain? And one of the reasons this really hasn't been done before is that it's incredibly difficult using verbal argument or conventional methodology to get quantitative information when you have these tightly synchronized, behaviorally synchronized groups. So what we've resorted to is using computational models initially to try to get inside the head of the individual to see how and why she behaves the way she does. Now, so I'm going to go sort of back in time to one of the first models of this type. And I'm going to sort of take us on a trajectory from our early understanding of collective dynamics to a very contemporary understanding, because it all relates together, as you'll see, especially the experimental tests of the theory. And so, at this point, all we could do, there is no available data. Not a dearth of data, there's no available data. So I assumed that individuals exist at certain positions. So individual I exists at centroid at time t, and of course they have velocities. They have self-propulsion. And all I assume is that when individuals get too close together, they tend to avoid collisions, they turn apart. However, if they become isolated, if they're leaving the group, then they exhibit an attraction towards others. That's this term here, and they may or may not also tend to adjust their speed to match that of their neighbors. And just these simple local rules, just mediated with near neighbors, can explain how these structures tend to form. But it doesn't give us much insight into the evolutionary drivers. So I mentioned, for example, that there can be attraction among individuals, or a tendency to align the directions of travel with neighbors. But I can't assess how natural selection would act upon those behavioral features, because I can't tell one fish, you exhibit more attraction than you, and I'll put you out in nature and see which one gets eaten most quickly. And so what we decided to do is to utilize virtual reality, whereby we could have virtual individuals that all look identical, but they start with random behaviors. They're not even social to begin with. And then we can use real predators to hunt these virtual prey and exert selection pressure on them, so we can then compact evolutionary time down to a realistic timescale. I mean, it still took us about 2 years. And this is just one example. So these are the virtual prey projected onto a translucent film within the fish tank, and we're looking through the film at a bluegill sunfish that's coming to attack. And it's very, very easy to see with your opercular openings which individual it's attacked. Now that individual, because we know the random number generator that we start with in the frame, we know what the phenotype was, the behavioral type was. And that behavioral type is less likely to reproduce. And running this over many, many generations with hundreds and hundreds of fish, you can clearly see that it's very dangerous to be alone, to exhibit no attraction and no tendency to align your direction with neighbors. However, it's not good enough just to be attracted to other individuals. This clump is also at risk. It's also not good enough just to align direction of travel with neighbors, because your motion becomes too predictable. What they actually evolved is a balance of these factors that minimizes risk, these virtual prey. So they evolved to group, and they evolved to move together, driven by this selection pressure. Now, if we look at these types of models now in three-dimensional space, regardless of the exact details of the model, and that's important, this model makes a fundamental prediction, that there are 3 states of matter within these types of groups. There's a swarm-like state, a bit like a mosquito swarm. There's this dynamic parallel state, which we sort of commonly think of as fish schools or bird flocks. And finally, there's this milling state, where the individuals perpetually rotate around an empty core. But of course, there's nothing in the equations that said go in a circle. This is an emergent outcome that wouldn't necessarily have been easy to predict just using verbal arguments. Furthermore, even if the individuals exhibit exactly the same social interactions, and we just vary speed, only speed, the interactions are identical, the social interactions are identical, we see the sudden transitions between these collective states. So when individuals move slowly, they become dense and disordered. As we increase speed, so what I'm doing here in the red line is I'm increasing the speed of the individuals a little bit, and then I run the model for a very long time to what's called dynamic equilibrium. And then I increase speed a little bit further, and so on. And what you can see is that if I look at the degree of collective rotation, the angular momentum of the system suddenly transitions from the swarm state to the milling state. But then it suddenly transitions at high speeds to the dynamic polarized state. Now, taking exactly the same model, what I'm now going to do is decrease the speed a little bit and then run the model to dynamic equilibrium and decrease it. And you may expect that it would show the same behaviors. Because remember, the social interactions here are identical, and the speed is identical here and here, yet we have two different collective behaviors. The system, the collective, has a memory of its previous group state, even though no individual has that memory. This is called hysteresis, and this is a phenomenon that you also see with physical systems. So this is liquid. You can see the bubble of air in this liquid, but it's cooled down to the freezing point. And if I tap it on the dashboard of the car, any imperfections in there cause crystallization, and ice spreads through the whole system. So it exists both as a liquid and a solid for identical interactions, in this case, identical temperature. This is going to turn out to be very, very important when we think about how collectives process information. So when I moved to Princeton to set up my lab there in 2008, I really wanted to not just do the modeling. I wanted to set up experimental systems whereby we could quantify the behavior of these animals in a sophisticated way. And so I turned to small schooling fish. These are called golden shiners. They're bred in the billions in the US to use as live bait by fishermen. So you can get 1,000 of these delivered to your doorstep by truck from Arkansas for around $80. So incredibly convenient. And on the very first day, we put them in a tank. This tank is about 2.5 meters by 1.2 meters. This is sped up, I think, 3 times normal speed. And you can see the fish exhibiting these transitions in collective states. Also, despite the depth of the water, whatever depth you give them, even in a lake that's very, very deep, these fish will swim at the surface. So it's a quasi-planar situation. And so in the years before deep learning, we developed some clever algorithms, which are completely superseded now, but nevertheless clever algorithms that allowed us to track the motion of all of these individuals simultaneously and not lose track of who was whom. And this is a group of 300 just focusing on one random individual. And if you recall, the prediction of the model is that regardless of the details of that model, the prediction is that there should be these 3 fundamental states of matter. So we filmed our fish for hundreds of hours, tracked them, and we've only ever seen those 3 states that we predicted. Furthermore, if you recall, we predicted that speed is going to be very important. And indeed, these are just 2 ways to calculate speed. As individuals speed up, the system polarizes, the system gains order. So when the system is slow, it's in the swarm state. When the system moves faster, they go into these different behavioral states. But importantly, the social interactions are not changing. Only speed is changing. Right? So, for example, we're collaborating with colleagues at Harvard who are doing manipulations of zebrafish, lots of genetic manipulations. And most of what they see in the changes of social behavior can be entirely attributed just to the knockouts affecting the speed of the individual. So it's quite a difficult challenge to associate social interactions in these types of systems. So this is another way of plotting it. This is real data. This is not a model. This is the rotation, the angular momentum against the polarization, and instantly you see those 3 hotspots, those 3 behavioral states, and the intermediate transitional states are unstable. The system will sit in one of these attractors and flip to another attractor and flip back. So in terms of the collective behavioral space, if you compare it, for example, to the mice that we saw earlier today, this is even though the system is complex, and these are complex vertebrates, the system itself has a very low-dimensional behavioral space, which is convenient. And so just to remind you, low rotation, low polarization is the swarm state, high rotation, low polarization is the milling state, and high polarization, low rotation, low global rotation is the polarized state. And as I mentioned before, we control, by the way, for edge effects and boundary effects and all that. That's why we need to collect so much data. Because, of course, the boundary plays a role, but we've excluded that from this analysis, and we can show that simply increasing the speed causes the system to transition. So when it's slow, they're in this state, and when it's fast, they're in this state, or this state. And locally, however, if we look at the local polarization, the fish do not seem to know if they're in a group of 30 or a group of 300. Their local world is identical. The density is identical, the packing fraction, the local polarization. We tried to train them. Can we train you to know you're in a small group or a big group? They seem not to be able to be trained to do this. We tried with zebrafish too, also failed.
Is the dashed line coming back, or?
No, there's two, I can't remember the exact details, but there were two ways to calculate this property, and so we just decided to show that regardless of how you do it, the results are the same. Okay, so working with Andrew Badal, who was a PhD student and is now a professor at the University of Washington, and Colin Tawney, who was a postdoc, now a lecturer at Glasgow University, we wanted to ask something a bit deeper about this relationship between speed and order. Can this help us understand how these groups, how the individuals within groups, process information? And so what we want to do is to set them an ecologically relevant challenge, and that is to climb a long-range gradient. So if you're a homing salmon, for example, you're climbing a chemical gradient. If you're a tuna, you're climbing thermal gradients. But those things are hard for us to manipulate. And what we realized is if you watch fish in a dappled light environment in a stream, they flit between the dark areas. Why do they want to be in the dark? Well, because they're cryptic in the dark from aerial predators. But that also makes the tracking extremely difficult, because we're projecting this light field onto the base of the tank. And so what you're seeing at the bottom is exactly the same experiment, but we've cut out the visible light, and we're lighting this purely with infrared. And so we can change the length scales and the noise parameters in our environment. So we can now track the motion of the individuals and look both how they interact with each other, but also now how they interact with a complex environment. And the light gradient is time varying. You have to keep finding the solution. And there's always one global optima and local traps, so to speak, local optima. And indeed, we found the first experimental evidence that as group size increases, the individuals become much better able to solve this problem. We thought this is terrific, because we have the acceleration data. So we can ask, over what scale are they responding to the environment? Over what scale are they responding to each other? Because the way we were thinking is what's called the classic wisdom of crowds. So this actually comes from Sir Francis Galton, who was a cousin of Charles Darwin. And he went to a livestock fair, and he knows a lot about lots of things. This guy was a polymath. But he didn't know much about livestock, and there was a competition to guess the weight of a dressed ox. And Sir Francis Galton realized that he had no chance to win. But almost 700 villagers had already entered the competition. So he calculated the mean of their estimates and was 1 pound off the true weight of the ox. And so the idea here is that what we thought was going on is that each individual is making a local, noisy estimate of its environment. And then the social interactions, they're not able to count like Sir Francis Galton, but the social interactions nevertheless effectively average those local inputs. So can we find evidence for this? Well, one of the cool things about doing experiments, not just doing modeling, is that the animals can do something wildly different. So while we found, of course, very strong evidence, as the blue line shows, that the social vector was extremely important, we found no evidence, whether you're in a group of 2 or a group of 256, that you are capable of estimating the environmental gradient over any length scale. So they're not integrating in time or in space. And so this leads us to a conundrum. If the fish are not capable or not utilizing the local light gradient, how on earth is the group able to climb the gradient? And of course, natural selection has found a very simple mechanism. It's a mechanism so simple that it's cognitively minimal, which could be advantageous, but it also, working with control theorists, we've been able to show it's also provably optimal. All the fish are doing is moving faster in light regions, the regions they don't like, and slowing down in regions they do like. Now that means you do spend slightly more time in the dark, but it only gives you around 10% benefit. It's very ineffective because there's no taxis. This is kinesis. And the taxis itself emerges via the social interactions. It simply does not exist at the level of the individuals. It is an emergent property that results from the network of social communication. Effectively, what's happening is, remember what happens when the group slows down? When the individuals slow down, they undergo this phase-like transition into a sort of dense solid state. But if they're moving faster in the light regions, they're in the fluid regime, like a living fluid. And it's actually the environment causing this phase-like transition within the group that allows the group to process information in the environment. And we've been able to show that this is evolutionarily stable for unrelated selfish individuals. And our little golden shiner fish, because this is a cognitively minimalist solution, a little golden shiner fish have inspired Shinobots, miniature robots, much like Radhika Nagpal's kilobots, that can implement these algorithms, because the algorithm is the result of the network of communication, not at the level of the individuals.
Sorry? Have you tried this experiment in an animal that is able to process phototaxi?
So, yeah, very, very rarely do behavioral experiments get replicated. This is one of the few examples where someone replicated this result with our golden shiners and found exactly the same thing. But they looked at a different species of fish and found that that species utilized this strategy plus the individual's processed information. So it's not mutually exclusive. So I was giving you a sort of suggestion that these relatively local, simple interactions can explain much of the dynamics we see, and that's true. But of course, these individuals are not automatons. They're not actually interacting via social forces. These models tended to come from statistical physics and chemistry, where thinking about forces makes sense. But in real animal groups, of course, I might be strongly influenced by you, but you not by me. And of course, they're not basing decisions on forces, but actually on sensory information. And so in addition to tracking the motion of the fish and estimating the body posture, which, as you saw with the mice example, is extremely important for deciphering the behavioral dynamics, we also calculate the positions of the eyes and use graphics processing units to raycast the photons onto the retina so we can reconstruct the visual fields for each individual. And so now for an arbitrarily chosen individual, this one here, you can see the left visual field in red and the right visual field in blue. This is all in 2D. We can now do this in 3D. And you can immediately see that this is a very complex, time-varying scene, where the individual is, as it moves, it changes the scene. As others move, it changes the scene. And so it's very tricky to understand how social interactions spread, because indeed, the sort of hallmark of collective behavior is that the change of behavior of an individual or individuals can percolate through the system and impact others, whether this is stress, fear, or in this case, an alarm behavior exhibited by the fish. This is work I did with PhD students Bryn Rosenthal and Colin Toomey.
Sorry, in previous image you were showing also where they are looking at? Or?
Pardon me?
In previous image.
In the previous video, the visual fields here, the left visual field is shown in red. So where these rays intersect with an individual, it can see that individual. If an individual is not covered by rays, it's behind another individual or individuals, and it's in the blind spot. Does that make sense? Because that's pretty important, because these fish are highly visual. And so here's some slow motion footage, and what we developed is a technique whereby we could trigger any of the individuals within the group to exhibit what's called a C-start behavior. This is a reflex behavior exhibited by fish when they're alarmed, predominantly driven by two giant neurons called mouth nacelles. And so 100% of the time when we trigger this behavior, we get a C-start from the individual. And so we know that that individual initiated this wave. And so we did 205 experiments where we know who initiated it. And you think, well, you're doing the same experiment, yes, maybe in different positions within the group, but you probably have a similar type of behavioral outcome. But that's not the case. If you look on a log-log axis, the number of events and the cascade size, actually most of the time we didn't manipulate the group behavior at all. Only sometimes did this lead to an avalanche that changed the behavior of the entire group. And this is reminiscent of other collective properties like news stories spreading on Twitter, tends to be a long-tail distribution, or power outages. So it's very hard to predict beforehand whether an individual is going to change the behavior of the group or not. And so to solve this, we had to do something that turns out to be extremely difficult. we needed to establish a functional mapping between the sensory input. Now, these fish are really visual. You can knock out the lateral line, nothing changes. Turn the lights down, they can't interact in any way whatsoever. And so if you look at this visual field, if this is time, and this is the left and the right eye wrapped around, with dark being an individual being close to me, light gray being an individual far away, and white being the boundary, you can see these sort of sudden transitions that's driven by the individual making these tail beats.
So the fish has phobia or?
So we've also analyzed the eyes of these fish. We did zebrafish as well. I was amazed that no one had analyzed the zebrafish eye. And we found that they don't have a fovea, but they have a higher resolution region approximately pointing upwards diagonally like this. Quite similar between the two species, which, I mean, I'm not sure how much we can really infer from that because they feed on the surface. So you need to see high contrast and predators come from above. So it could be very much for anti-predation rather than social behavior. The rest of the eye is very homogeneous. So we want to then understand how the individuals integrate their past behavior and this social information into a decision. And we've deliberately chosen a very low-dimensional behavioral output. Remember, it's driven predominantly by 2 cells. So the brain has to have evolved this dimensionality reduction. Yet this is super important. This species has a 92% mortality per year through predation. Right? So this alarm behavior is very behaviorally relevant. And I won't go into the details, we don't have time, but we really did need to do this computational visual field reconstruction, body posture mapping, as we saw with the mice earlier, and machine learning techniques. Okay, so these techniques are always changing. There's new techniques coming all the time, and this is a really exciting time to do behavioral biology. But I won't go through the details of what we used. I think the details don't actually matter that much. The new techniques would find the same types of things. And so we worked out which features they're paying attention to. And surprisingly to me, they're not paying attention to derivatives. They're paying attention actually to the 2 simplest visual features they could pay attention to, potentially again for resilience and robustness in the face of turbidity, for example, in the water. And this allows us then to reconstruct what we call the hidden network of interactions. So if you're a neuroscientist, as almost everyone in this room is, and you want to try to understand the behavior of a circuit in the brain, you can go in and look at the functional connectivity and manipulate that. But when we're looking at social circuits, the social interactions are invisible. They have no physical form. And so this type of technology allows us for the first time to reveal these hidden networks of interaction that give rise to collective behavior. And this is not just, oh, this individual is close to this one. This is based on the visual information coming in and the algorithm that we've determined that they use when they process that visual information. And these edges actually represent quantitative probabilities. Okay, so this is a predictive quantitative network. And even though they use the simplest visual features possible, because of the geometry of the system, some individuals may strongly influence others, but not be influenced by them, and vice versa. So we have a weighted, directed network of communication of this behavioral spread.
Sorry, influence, do you mean visual?
Influenced behaviorally. Everything is visual. Visual is the sensory modality, influence is the behavioral response. Yeah? I mean, it need not be visual. We could be thinking about olfaction or acoustics, but in this case, it's a visual system. That's the sensory modality they use to understand what's going on around them. And so I'll just show this video again, because this is the first time we've ever been able to reveal this network. And it's not a small world network or a Obergurgl network or a scale-free network. It's not like any networks that we've seen in the models.
So if it's purely visual, what happens if you look at a swarm in the dark?
They don't do anything in the dark. They can't see each other. They can't do anything in the dark. We can do that with infrared light. We can even put the light down to low levels that we can still see them, but they can't see each other. Yeah, they can't do anything in the dark. And so I don't have time to go into it today, but if you look at this network connectivity, we're able to show that the structure itself actually minimizes correlated information within the group. We also think the fish are acting much like coincidence detectors in a neural network, where depending on the time and the spatial information coming in, they respond in very different ways. So I just want to say something simple about the social influence and network structure, something quite basic that's kind of surprising that we didn't know about. Because it's often assumed that if we look at a network, a social network like this, the most socially influential individuals are going to be those with many connections to others. This is called a high degree in the network literature. And whose neighbors have a low cliquishness, or a low clustering coefficient, to use the network terminology. Why is this assumed? Well, this is assumed because until now, people believed that behavior spreads much like a contagious disease. And they use the same model. They just make it go faster. Right? And if I have a disease, I don't believe I do, but if I had a contagious disease, the more of you I interact with during this meeting, the higher is the probability I will infect you. However, if I only interact with the gentleman at the front here, and they don't want to interact with the riffraff, with the rest of you, they're a little clique, then I could infect them, but because they're a clique, I can infect this whole little clique, but it's not going to spread out. So this makes intuitive sense, and this is true of disease. But no one has ever checked and asked, because this is in tens and tens of models of behavior, has asked, is this actually how behavior spreads? And so we can ask that of the real data, not guess, not model it, measure it. And what we find was no. It turns out that the most socially influential individuals are those with a low degree and whose neighbors have a high clustering coefficient. Again, the experiments show exactly the opposite to intuition, which is why it's so critical to do this type of work. Not just to guess how the world works, but to measure how the world works. And of course, the important thing is we can assess why. And there are some details to this, but broadly speaking, it's because behavior transmits as a complex contagion, whereas disease is a so-called simple contagion. What do I mean by that? Well, let's say I have a disease again. The more time I spend with Florian at this meeting, the higher is the probability that I will infect him with my disease. But let's consider behavior. If I have an opinion, say about Donald Trump, and I try to persuade Florian all day that Donald Trump is a great president, I may not be able to persuade him. I hope not. Probably not. Well, Trump, of course, is too polarizing. But what I mean is that behavior doesn't, you just don't have that continuous probability of adopting the behavior of another individual. But say I convince Florian a little bit, and he goes closer to threshold, but he hasn't crossed threshold, he hasn't changed behavior. But then I influence you, who influences you, who influences you, who influences Florian. And then you change him. I've influenced them both directly and indirectly through this loop. And that turns out to be absolutely critical, because whether we look at fish, birds, or humans, they all seek reinforcement from multiple opinions before making a decision. Right? And you also seek to decorrelate information. I was in New York City one day, and my friends were trying to persuade me to buy an album. I wasn't convinced. But when I sat on New Jersey Transit to go back to Princeton, the couple in front of me was talking about the same album. That's uncorrelated information. It's very unlikely they got that information from my friends in New York. And so I bought the album. And this is something that we do all the time. The fish are doing it in a kind of a cool way. They don't know who's whom, but they respond to different parts of the visual field in interesting ways. Okay, so now we can go back. So we have this new understanding of how network connectivity impacts social contagion. Yeah.
Right, so in the example that you gave, you identified two possible directions of influence. I still cannot understand why low connectivity would be with more influential.
Right.
Right? Because as you can influence Florian, you could influence with everybody else, and then they could be influenced from all other sources.
You're absolutely right, and that need not be the case. They've evolved such that few strong connections is much more influential than many weak ties. And this is exactly what Damon Centola has since found. There's a famous paper called The Power of Weak Ties, but Damon Centola published a paper in Science a few years ago. In fact, our Fisher on the front of his new book, because he found the same thing in humans. In actual fact, we all think of these like many, many weak ties being very important. Turns out not, in terms of changing behaviors. So the fish are very much like humans in that case.
So just because of this example
Yeah.
that we're giving with the record, we also can choose, sometimes when we want to make decisions, we also can choose the peers that we want
Yes.
to. So do you have
Yeah.
any evidence?
I'm going to come to that later with baboons, because these fish are dumb as anything. They can't, sorry, zebrafish. Zebrafish, of course, are very smart. But these golden shiners, I mean, they really have evolved to do everything as a collective. And they live in a fission-fusion dynamic where they're mixing and merging with hundreds of thousands of other individuals every day. So in this case, I don't think it really matters who's whom. What does matter is, related to the previous question, what really matters is spatial position in the network. The edges of the group, because you have stronger but fewer connections because there's no one else on the outside, those edges actually are much more likely to have the information flowing in from the edge.
So they are all the same gender?
No, this is mixed. You cannot tell until they go. They're not in breeding season.
It doesn't matter if the neighbors are more...
No, we can't tell. No one can tell. These are actually juveniles, so they don't breed anyway at this age. But were they to breed, they can grow a bit bigger. Were they to breed, they only do that once a year. And then it's driven by temperature, and they change colors, and they change behavior. They do all sorts of cool stuff. But for the rest of their lives, they behave like this. There's no way, you cannot distinguish behaviorally between males and females.
Do you see if they have a tendency to go toward the center of the group?
It's a great question. I've got a whole bunch of slides that I took out for time. But basically, to sort of summarize this, there's a sort of classic paper by William Hamilton called The Geometry for the Selfish Herd, which people miscite as geometry of the selfish herd, and I can always tell whether they've really read the paper as to whether they've miscited it. And this argues that you should always try to get to the middle of the group. But, and biologists love this theory, but no one's really tested it. And what we actually find is that doesn't have any dynamics. If you have the dynamics and the social connectivity, the best place is to be at the front and the edge, because you have the best personal information coming in from the outside and the best social information. So we've tested that in the field with sonar, a new type of sonar, and actually showed that indeed predators specifically attack the back of the groups, the informationally weak spot. So what you don't want to be is at the back. And if no one wants to be at the back, what happens? Well, the group starts moving. Right? So these groups are on the move all the time because no one wants to be at the back, not driven by cannibalism, but by predation.
So somewhere you said high clustering coefficient.
Yeah.
And then
The high clustering coefficients really...
These concepts are from a small group.
No, they're just from network theory, graph theory. These concepts have been around for 100 years. But the clique is important. This tight connectivity, the clique is important, because remember when I influenced Florian, I could influence him directly, but influence him also through his neighbors. So if I'm connected to Florian's neighbors, then I can influence him in two ways. If I'm not connected to his neighbors, I can't do that. And that's why the cliquishness is important.
So what do they do at night when there's no light?
They go stationary. They find somewhere to hide, and they just become completely stationary. In the lab, they just distribute randomly, random positions, random orientations across the whole tank. Yeah, they go into a sleep-like state. And so now we can do a whole new bunch of experiments where we trigger the individual, but now we freeze time. At the moment we trigger the individual, we freeze time, we reconstruct the visual fields, we use the algorithm that we know that they use, we work out the probabilities, and we now predict who is going to be infected by behavior, when, and how far will it spread. And it turns out even though this is a real, complex, noisy, real biological system, I'm astonished at how well we can now predict whether changing an individual, triggering an individual will lead to a cascade or nothing. And so now we can really predict what's going on within this group. But this is really hard to do. And the reason this is so hard to do is because of causality is very hard to infer. Right? If I suddenly jump up, and then one of you jumps up afterwards, it's such a rare thing that it's probably me that caused that. Right? So these fish don't alarm very often. And so if one alarms and another alarms in close second, I can infer causality, that first alarm caused the second. But what about when the third one or the fourth one did? Are they interacting just with the most recent one they've seen? Do they somehow integrate over time? There's lots of complexities here. And so to try to get around this, to try to control causality, we've worked to develop a holographic immersive virtual reality for these organisms. And this is work with Andrew Straw, John Stowes, and Max Hofbauer. And so this object is not 3D. I'm just going to tell you that right now. It's not a 3D object. It's a 2D object. But your brain believes it to be 3D. This is such a powerful illusion that I can even tell you that you're being deceived, and you'll see now it's broken. Now, of course, when it goes back into position, not even in exactly the same position, and it will pop back into 3D. It has a volume above the table in your brain. So that means we can have objects. This shoe is not 3D either. So we can now have objects in the tank, so virtual zebrafish in this case, in the tank swimming and interacting with real zebrafish using this illusion. Because here's a virtual fish below a real fish. We actually use juveniles, as I'll show you in a moment. So we project this onto a curved dome. It's about 35 to 40 centimeters. The fish are less than a centimeter, about 21, 22 days post-fertilization. We then can control for all the distortions. And in this particular experiment, we put an artificial pillar in there, a visual pillar. Pillar doesn't exist. But this looks weird to us. But remember, from your perspective before, that illusion looked very, very real. And so indeed, the fish will avoid the pillar, even though it's not there. So we turned to working with zebrafish because they're cool. And ultimately, what we would like to do is collaborate with people like Florian to try to understand the implementation of the algorithms in social behavior. And I think we're getting closer and closer to being able to do experiments with very young individuals. Their social behavior does kick in quite early. And so this is a real zebrafish. We've tracked the body posture. And this is our photorealistic virtual zebrafish. And so what I'm now showing you is a virtual fish that's following an unrealistic trajectory, just a circle. But just to show you, the real fish is interacting with it in full 3D space, even though it's not there. This is a complete illusion.
How old are they? Or is he?
21 days. We studied them between 21 and 24 days. I think you could go younger. They schooled 90% of the time at this age. But remember this, so we can put in as many virtual fish as we want. We can put in pillars or plants or whatever we want, predators. But the world only looks correct from that real fish's perspective. If you put another fish in there, another real fish in there, the world would look weird from its perspective. So that's a limitation here. But what we've done is we've developed 6 of these systems. This is just looking from below, and we can then network them. So each individual can interact with a holographic representation of the other individuals, the other real individuals. So we call this the matrix for obvious reasons. So we now have 2 fish that are not in the same physical world, they're only in the same holographic world, interacting with each other. And the behavior is very, very similar to the behavior of real fish. Here we have 4 fish interacting in the same holographic environment. And of course, we could have virtual fish in the loop here as well. I mean, so it's really up to the imagination when you start working with these types of immersive environments. This is I just pulled this from a website earlier today. Just shows a bit like we saw with Robert's talk with the mice. You can then superimpose hundreds of individuals performing a behavior to look for patterns. In this case, they are disassociating and then reassociating with the virtual individual. And you can see that when they're interacting, they tend to occupy the same vertical dimension as the virtual fish. And they do this in reality. And so the advantages here is that we control precisely the visual experience. We can have exactly the same sensory inputs to the same individual or to multiple individuals. It allows us to gain much deeper insights into how the brain makes movement decisions. This is actually, I'll come back to this in a moment, this is actually real data for how the individual brain makes decisions in this scenario. And also we can employ this sort of social patch clamp approach where we could have, once we think we know what the algorithm is they're using, we can test that directly in the virtual system by having the virtual organism, employing that algorithm in the loop with the real organisms. So this allows us to much more quickly test our ideas. I'll come back to that later on. So in the last part of the talk, I want to talk about leadership and collective decision-making. How do grouping animals make informed unanimous decisions? And then I'm going to sort of turn it around and see whether we've actually learned something about how individual animals make decisions. And so I'll start again with some old work because it sets the scene for some very recent experimental tests. So as we all know, fish are very smart organisms and they have very good memory. So for example, this individual here knows there's food over there. The others don't know that. So it has goal-oriented behavior. Or perhaps it recognizes there's a predator over here that the others haven't seen and it wants to move away. So what we're assuming is that the individual in its brain has a goal-oriented vector. Now, does it have to signal to others, I know what's going on, follow me? Do others need to recognize who has information and who doesn't? Well, we have no evidence of this. So I turned to a simple simulation model, just in 2 dimensions. And I made the assumption, which we've now got very good evidence for, that individuals reconcile their social tendency with this goal-oriented tendency with a continuous weighting term that we call omega. Now, if omega is very low, this goal-oriented behavior doesn't play much role in their dynamics. If omega becomes very high, then it can be dominant, and of course the individual may leave the group. Now, importantly, I'm coloring in the informed individuals in bright colors. That's so we can see who they are. In the model, there's no cheating going on. They start at random positions, random orientations. There's no way to infer the state of other individuals. I'm not saying that's true, but that's the evidence we have suggests that's true. And you'll see we don't need to have more complex assumptions. And so we can ask, if we have a preferred direction in these informed individuals, can information be conveyed without signaling and without individual recognition? So we have just 1, 5, or 10 informed individuals in a group of 100. And the green cross just shows you the centroid of the group. 1 informed individual isn't able to convey the information. 5 informed individuals, after an initial reorganization where they occupy the frontal edge, you can see they're kind of going in the right direction. It's not super accurate. But with 10 informed individuals, suddenly and spontaneously, they move very accurately. And so we can quantify this. This is a normalized accuracy, 0 being no information transfer, 1 being perfect information transfer. And I should note, in all of these models, we have noise, because the sensing systems are noisy, and translating that to motion is noisy. And so you can never get all the way to 1 because of internal noise in the system. But what we see is, regardless of group size, as you increase the proportion of informed individuals, you increase asymptotically the accuracy. So it's very fast to begin with, and then plateaus. And if we choose, say, 85% accuracy, and we look at small group, a group of 10, we need around half of the individuals to have that information. But as we get to larger and larger groups, so here's a group of 200, we now need less than 5% of the individuals to have the information. Yet to the observer, to the biologist observing, it looks like everybody knows what's going on. Everybody knows where to go. But that may not be the case, and often gaining information in the natural environment is very dangerous. And so you expect a frequency dependence. We've shown this in evolutionary models. We've also tested this experimentally, again with schooling golden shiners. The red ones have information about a green laser. Normally they have no response to a green laser. And we can then guide them around the tank. But you could see the wave of information transfer crossing the group. And indeed, as we predicted, we have the steep slope, and that flattens out, that plateaus.
Was the green laser good?
The green laser, they have 0 response to a green laser normally.
No,
but if we,
in that case, was it good or bad?
It was good.
It was good.
In this case, it was good. We've also trained them to bad. Didn't work so well. So these are actually automated feeders, and it's random. The laser's on a gimbal, and it just zaps randomly which one. And then, of course, when we do the experiments, there is no food present. But in the training, it's all automated. But so far, all of these individuals have been in agreement about what they think the best thing to do is. But in many cases, as I mentioned before, these individuals live in fission-fusion societies or systems. Just like us, they have different sources of information, different knowledge, and different contexts. So can they somehow integrate that to come to collective consensus decisions? Now, it's not like we can really make many decisions about what we do this evening, but if we were in a town with bars, we could, for example, ask, like, let's go to a bar together. Maybe 6 of you have an opinion about one place, 5 another, and the rest of you don't know. I would ask the question, you put your hands up, I calculate it, and I'd pass back the consensus. But they can't do that. So can these individuals, without any ability to count, come to consensus decisions? And so what turns out to be important is the number of individuals with a preference, the strength of the preferences, but also the geometry of the problem itself. And so I'm going to keep one subset within the group to prefer 0 degrees, which is along the x-axis. So they're always the lower dotted line. The other group may agree with them. That's the model I just showed you. With 0 disagreement, it's the same model. But now they may disagree by, say, 5 degrees, 10 degrees, 20 degrees, all the way up to 180 degrees, where they actually want to go in exactly opposite directions. They start at random positions, random orientations. You don't know if anyone agrees with you or if anyone disagrees with you. And so here we'll have a group size of 100, with 5 want to go one way, 5 want to go the other. And for each angle, I run thousands and thousands of simulations, and I show you the probability of where the group goes, and they all go together. So you can see below a critical difference of opinion. They split the difference and spontaneously go the average direction. But as soon as they reach this critical bifurcation point, this is a pitchfork bifurcation, they will then come to a consensus regime where they either go one way or they go the other way. It's equal probabilities. The group's not splitting. But what if I now, instead of 5 versus 5, I have one extra individual in one of those subsets, so 1% of total group size.
So the point of bifurcation can predict, is it all the same? Or
Excuse me?
the point of bifurcation?
I'll come to that a bit later. The angle? You mean the angle at which they bifurcate? This is the angle here.
Distance or...
No, this is the angle, degrees. This is the angle at which they bifurcate.
Oh, I see. Yeah.
Right, so if I just show you this again, when the angle between these two options, this angle here between the two options is below, in this case, around 125 degrees, they are in the averaging regime. As soon as they exceed that, and of course, if you're actually moving towards a red and a green target, as you get closer, the angle is going to increase, so you're always going to enter this consensus regime. Then the system will switch from averaging spontaneously, nothing changing in the brain, nothing changing in the model, but they'll spontaneously switch to a consensus regime. And so if I now have 1% extra, random orientations, random positions, you don't know if anyone agrees with you or disagrees with you, now below that critical angle is exactly the same, they choose the average, but now above that, over 95% of the time, they will spontaneously select the majority direction without being able to count.
Do you think nothing changes in the brains of the informed individuals? If they're either forced to go to the place where they don't want to go, or to the ones where they...
That's a great question. So in the paper, I put in a way that we could move, so it relates to the previous question too, move the bifurcation point. The way you can move that is to have a sort of forgetting parameter. So if you end up going the way you don't want to go, you just begin to not have a preference anymore. That allows you to move the things. I thought it didn't do much else. Turns out it does a ton more, and I'll come to that in a moment. That turns out to be absolutely necessary when we compare it to the real system. So if we look at these models, you can see the sort of local noisy nature of the interactions. There's an equal number that want to get to the white and the red, and so it's going to be random. In this case, they choose the red. And if I let this run longer, they will bounce back and forth between these two options.
That's a simulation.
This is a simulation. Now I've added one extra white individual, and almost always they will go to the majority preferred direction, even though the interactions are local and noisy. Now, there is a regime, if omega is large enough, and they're unwilling to give up on their preferences, related to what Florian just mentioned, where they enter a regime where they can no longer come to consensus. So this forgetting term actually prevents this from happening too. And so I I wanted to work on other things for a few years, but then I sort of regained interest in this when I tried to evolve what was going on. Because the way I wrote it in this paper, I was so rushed for time by the editors, the way I wrote it is, isn't it remarkable that they can do this despite all of these uninformed, unbiased, noisy individuals? What I didn't realize at the time is it's not despite them, it's because of them. You need that population, a sort of recruitable population. You don't just have this direct lateral inhibition, you're doing it via a third subset, which mathematically turns out to be very important. And so I wanted to think about also the fact that, there's conflicting interests. These are selfish individuals, right? So if we have, in a human decision-making scenario, if we have a conflict, say, about whom to hire in a business, and you can't come to an agreement ever, this is detrimental to your business, right? If animals can't decide whether to go to that food patch or that food patch, predators are going to come and take them in a moment. It's really important that they make accurate but fast decisions. And they're dealing with conflict. Again, you can sort of think about this in the brain. You have sort of conflicting neural groups that inhibit each other.
So those that they are always at the edges of
The informed individuals will spontaneously occupy the edges, the frontal positions within the group. And that's exactly what we saw with the real fish when I showed the video with the green laser. They will move to the front and then guide the group.
Yeah, I would say they are always at the edge, or almost always the same fish they are at the edge. I want to say if there is a weakness point at the middle of your population and the strongest point at the side.
No. So these just the position you adopt simply is based upon your informational status.
Yeah.
I mean, you nicely explain how are they doing that, but I'm not quite sure why are they doing that. So what's the advantage in staying
I'll come to that. I'll come to that later. That's why I was doing the evolutionary models.
So, but when you talk about informed individual, and when we talk about those guys in the edges, so what type of information, like a predator?
I don't care in the model. All they have to have is an internal vector to where they want to go. That could be part of our migration route. It could be that there's a mating opportunity over there. The model is agnostic to that property.
Have you looked at the models also with predators as opposed to repellent as opposed to attractant? Because intuitively it seems that if you have two predators coming to a group, then this bifurcation is not so good, because at some point, I mean, you want maybe to keep in the middle rather than choosing going away from one but towards the other. So
yeah, so I mean, you're hitting on a really important point here, that predators have evolved to exploit these rules. And so you've got everything from bubble netting, where whales will put a net around the group and force them in. You know, when you see these swirling bait balls, that's when the predators have been successful. They cut the silver sides or the sardines from the main group, and they're doomed. It's like, you know, if you cut off cells from your arm and put them under a microscope, they'll still move around, but it doesn't mean that they're, you know, going to survive. And so what predators, and so I didn't show this, but I mentioned to you the study that we did of real predators attacking the groups in the wild. And we were sort of interested initially in the collective behavior of the prey until we realized, oh my goodness, the predators are also hunting collectively. In this case, the predators formed lines that allow them to slice through the groups. And if they do this, if the lines are long enough and they can slice the group and they can't reform again, then you've got two small groups. And if you look at the risk, like how close they get to the mouth of the predator, it dramatically goes up below a critical group size. So the predators are hunting in teams effectively to slice through these groups, to make them small, to reduce this information percolation. So there's lots of other stuff going on, you're right. And we have looked at this in terms of predation. And so, you know, I said that these are selfish individuals, genetically selfish individuals. So what's to stop these individuals who are in the minority just from becoming more strongly opinionated, having stronger preferences? Can they then exert their influence, going to where they want to go, over the other members of the group? And so if we have 6 versus 5, and I fix the majority preference arbitrarily to 0.3, I'll show these numbers, exact numbers don't matter in a moment. And we have the minority, if it has the same preference strength, as I showed you before, the majority will win around 100% of the time. But if the minority becomes more and more strongly opinionated, they can begin to exert an influence on the group. 0.5 means half of the time the minority wins, and half of the time the majority wins. But if the minority becomes sufficiently strongly opinionated, they can indeed dictate the motion of the group, although this comes at the massive cost of groups often splitting, as I showed before. But this is the case where everybody in a group has an opinion. Now, in nature, because of the fission-fusion dynamics and other reasons, there may well be individuals that don't know what's going on. Or individuals that know what's going on, but they don't care whether they go this way or this way. Now, normally, when you sort of think of this, you think intuitively, well, this is just going to add noise. It's not going to change much. It's just going to make the results noisier. Absolutely not. They have a profound impact. And so here the minority has a stronger preference than the majority, 0.38 versus 0.3. And now we're looking at the percentage of uninformed individuals in the group. And if there are no uninformed individuals, the minority's preference is strong, but it's not that strong. The majority still wins 60% of the time. But if I put in 10 or 12 stupid individuals into the group, this pushes control further to the majority. Of course, if everybody is uninformed, then it's chance. But I can increase the preference strength of that minority further, and further still. Now the minority will win 98% of the time, but if you have around 12 or 15 unbiased individuals, or individuals that don't care what's going on, they return control spontaneously to the majority. They spontaneously democratize decision-making. They also act like a social glue that prevents these opposing sides from ever being able to see eye to eye. They act as a conduit of information through the system. And so we asked ourselves, well, is this a general principle? This could maybe explain why we don't see these types of arms races within the natural systems. Because there are many systems in nature and technological systems where local components can influence others and be influenced by others. I mentioned technology or neural dynamics or social dynamics. They all have this feature that you influence and are influenced by those with whom you interact. And so we can now create a very simple model. We take space out completely, and we just deal with a network. And now the colors are just representing arbitrary opinions, and the edges are going to represent interactions between the individuals. So this one thinks red is best, this one thinks green is best, and they each have a small probability of changing each other's opinion. In this case, nothing happens. But of course, as I mentioned before, there's this concept of complex contagion, where you're seeking reinforcement from multiple individuals. And so now if you interact with multiple individuals, if the probability of being convinced by one of these green individuals was Q, the probability that red is convinced now is greater than 2 times Q. That's all you need mathematically in this model. And then this increases the probability, it's not deterministic, the probability this individual will change state, will change behavior. Now that's not very interesting for 3 individuals, so we simulated 10,000 individuals. And this was work I did in collaboration with Thilo Gross and Gervin Demirel at the Max Planck Institute for Physics of Complex Systems. They're now in Bristol University. And I kept them blind to the predictions. And when they sent me this graph, they were very confused. Because if the majority and minority in the network have an equal strength of opinion, the majority wins, as you'd expect. If we increase the opinion strength of the minority, they become intransigent, unwilling to give up on their opinion. Then the minority can win if they're strongly opinionated enough. But what about if we have now a proportion of nodes in the network that don't care or are uninformed? Then initially nothing changes, but then we reach a tipping point where a tiny proportion of individuals that don't care what happens actually dictate what happens. And the system flips to majority control state. And because this is a large system, a very large system, this is why we have this very strong nonlinearity and a very sudden switch in the dynamics. And the cool thing about this model is you can use moment expansion and moment closure to actually mathematically prove this result. And really, by we, I mean they, Thilo and Gervin. This is well beyond my abilities. But the nice thing about the mathematical model, so the simulations are shown as circles, the mathematical analytic model is shown as solid and dotted line. You can see there is only one solution below that critical density of uninformed individuals, and that's for the minority to win. But then, as we increase the density of uninformed individuals, there's an appearance of what's called a saddle node bifurcation, and the system flips to the stable majority state. There's also an unstable undecided state. And the cool thing about this is now we can look at the 2 things that really matter, the strength of that minority's preference versus the numerical advantage of the majority. And we can deal with any system size. Again, you see here, if everybody has a preference, the minority can win by being intransigent. But now I'm going to show you the region that flips from minority control to majority control once we reach that critical density of uninformed individuals. And I'm going to show it in white. And it's massive. So now the majority wins in the white and in the gray. The minority can only dominate here. I'm not saying this is a good thing or a bad thing, but this is inherent in the system dynamics. And so we think this is a general principle. We've found this in a whole series of papers, some of which I'll mention in a moment. Coupled oscillators, neural systems, social systems. We find this again and again and again. We find that the small proportion of unbiased individuals increases the probability that consensus can be reached. What's really cool, they also massively increase the speed at which decisions can be made and the sensitivity of decision making. So if you imagine, this is kind of cool because it means your nodes, whether they're neurons or organisms, can be quite noisy. You know, maybe it's hard for natural selection to really make them very good at their job. How do you make the system as a whole very accurate and very precise and very fast? You add in more noise. You add in these uninformed, unbiased individuals into the circuit, and that should spontaneously make the circuit very fast, very accurate. And so we've been able to then go back to physics and look at physical spin systems, how magnets work, basically, and say, what if the magnetic particles have a bias to go up or down? We reconstruct all of the same results. We can, even working a recent paper with Nir Gov and his group at the Weizmann Institute, we've been able to prove the result with mobile animal groups. This is nice, and this is just showing differences in system size. You go from this continuous to a discontinuous transition. But what I'm really interested in now, and this is all unpublished and it's very hand-wavy, is does this actually tell us something about how individual brains make decisions. Anyway, I just want to finish with actually testing the primary prediction from the model when dealing with animal groups, which is that, remember, the uninformed or unbiased individuals should inhibit the influence of a strongly opinionated minority and return control to the majority. They should effectively democratize decision-making. And so turning to our little golden shiners, with Christos Wanu, who is a postdoc in my group, is now a lecturer at Bristol University. And Christos is a brilliant experimentalist, but I wanted to keep him blind to the predictions, right? So he doesn't, accidentally find something that maybe he thinks I want him to find. And so we trained the fish, or he trained the fish, to have preference for a blue target or a yellow target. Fish, zebrafish too, have an intrinsic color-based learning bias, whereas for the same amount of training, yellow is just like crack to a zebrafish or a fish. They just learn much better. So those are going to be our strongly opinionated individuals, and those to the blue are our weakly opinionated individuals. And we can even run these experiments without the targets and so forth. And the model predicts, going back to the fish schooling model or flocking model, that really we should see a strong effect for a small absolute number of uninformed or unbiased individuals. And in our experiment, if we have no uninformed individuals, the minority, because they're so strongly opinionated, they dominate. But if we put in 5 or 10 untrained, uninformed individuals, exactly as we predicted, the system flips back to majority control. So there's something, again, counterintuitive here. Because diversity of opinion strengths, and particularly having individuals with weak opinions, spontaneously democratizes the decision-making process and prevents that extremist minority from taking hold. But remember, these uninformed or unbiased individuals are participating in the decision. In many human systems, humans that are uninformed or unbiased are removed from the decision-making process. I often think about this in terms of like jury decisions or other decisions, whether we could sort of test this with humans. We may try and do that. And so talking on humans, this type of work, especially when Trump was elected, we really started thinking about polarization within human systems. But coming back to the more natural world, my final example, I want to talk about baboons. This is work I did with Ari Sternberg-Preskin, who was a PhD student in my group, who's now a junior professor in Konstanz, Damien Farine, who's now a PI in my department, and Meg Crowfoot, who was at UC Davis, is moving to Konstanz to become the 3rd director of our institute later this year. And so we fitted GPS collars and accelerometers to almost all adults in a wild, unhabituated baboon troop. This is an incredible amount of work. Sample size 1, because we've only got one group, but when you realize how much work this is, you'll appreciate it. Of course, for ethical reasons, we did not put collars on juveniles. And so when you look at the raw data, every second we now have the location of these baboons in Kenya. And looking at the data, I found tens of thousands of examples where some individuals tend to go one way, other individuals tend to go another way, but much like our fish, they will almost always stay together. And these individuals give up on their opinion and rejoin the group.
What are the colors here?
The colors represent whether they're male, female, or subadult. And so what I was kind of hoping to find was that, people, when they look at, so this species has a very strong, very linear dominance hierarchy. All males are dominant to all females, and the dominant male is dominant to all the other individuals. So it's a linear hierarchy. That hierarchy is based on aggression, who can get access to mates and all this kind of thing. And so I've always thought it's ridiculous when you watch TV that the dominant male is telling all the others where to go and what to do. I thought perhaps there's a hidden hierarchy of influence in the group. Turns out not. But it also turns out that the dominant role plays absolutely no role at all in terms of movement decisions. Whether you're a dominant male going this way or a subordinate female going this way, it's 50-50. It doesn't play any role at all. Now, that could be because of uninformed individuals. Remember? The strength of preference can no longer manifest itself. Or it could be that the dominant individual is, the system's evolved such the dominant individual is perfectly happy for this to be democratic, because when they get to the resource, ultimately, he can dominate it. But the cool thing is we can now reconstruct these bifurcation diagrams, so 10 years afterwards, with data from wild primates. And we find this very strong evidence for this bifurcation that we predicted. So even though that model is deliberately simplistic, it's not trying to capture all of the details, the results are so robust that we can find it in a wide range of different contexts, potentially even within the brains of individuals. But of course, these baboons are not fish moving in an empty-ish environment. They're living in a complex vegetated habitat. This looks like a picture, but it's not. This is a three-dimensional point cloud collected with this drone. That's me and Ari and Damien in the field in Kenya. And these drones can now get accuracy of around less than 1 centimeter spatial resolution in 3D. And they automatically do everything. Just throw this thing into the air. An hour later, it comes back with these data. You can see the tire tracks in the roads. You can see the trails. It's really, really exciting. And so we can now put these high-resolution GPS data into the environment in which these individuals have evolved. We can then do the visual field reconstruction in 3 dimensions to look who can see whom, and so forth. And so we could then create a preference landscape for each baboon, because these animals have memory. They know where they've been before. They have preferences, and at certain times of day, we know what those preferences are. In the evening, they want to get back to the sleeping tree. In the morning, they want to go out and forage. And so we can ask, how important are your visible neighbors, the habitat you've previously used, and so forth. And what's really cool, again, we got a surprise, because they're not really interacting with each other as points in space as we thought they were. They're interacting with each other probably via pheromones, chemicals, because like ants, they're basically creating trails through the environment, and others tend to follow those trails, even when they can't see the individuals. And those trails have a life of around 4 to 5 minutes. Right? So these animals are probably, and of course this allows you to come to consensus, because each individual is contributing to the trail. Much like ants can choose between options, the baboons are doing something very similar. And so this is a nonlinear axis. This is how well can we predict precisely where each individual is going to go over around 5 meters. And you can see that with a habitat, of course, they don't want to go through trees. We can get a certain level of description with social. We get more. But if we combine this together, we get much, much better predictive power. So it's a really exciting time to start studying animals in their natural environments. I think it's very powerful to study animals in the lab too. I'm not dismissing that in any way. But I think it's also important to try to understand how animals move. And many organisms, such as this is real data from migrating birds, are actually migrating over much of the planet. And these organisms are particularly susceptible to climate change. So we want to use these birds as an array of sensors to tell us about the health of our planet and also to protect them. And one way we're trying to do this is Martin Mikulski, who's the other director at our institute, has developed a technology called Icarus, which was put onto the International Space Station in August, which in principle, if it works, it's an experimental system, will be able to collect the data back from the tags. Because the problem is, you put a little tag on the animal, and you don't have enough power or weight to put a cell phone thing on it, or perhaps the animal moves into areas where there's no cell phone coverage. How on earth do you get the data back? So here, the International Space Station covers most of the globe and can relay the data to Germany. That's the idea. And this type of technology, this is not using Icarus, but this type of technology, these are real data, by the way. We've, of course, put the 3D animation on top of it. But we can have every wing beat of these birds as they're soaring in thermals. These are storks. And we can make predictions, and we can ask questions about leadership. And we find, indeed, as we predicted, there's a small number of individuals, 6 individuals, that guide the others where they're going each day. And they have to find these thermals, and they also have to decide when to leave a thermal to look for another thermal. And so there's all these challenges that these organisms have to face. And they have to do this as they cross into sub-Saharan Africa. And it's kind of amazing to me that now we can get the gliding and flapping flight from the accelerometry data. This is real data. We can actually use the fact that there's physics involved with flight to actually look at the distribution of wind within these thermals in ways that even meteorologists can't get this level of detail. We can then look at the mesoscopic decisions as they move over tens of kilometers, and we can track them over their entire lifetime, and did track them over their entire lifetime. And one of the cool things we could do is we can predict who's going to be a leader or a follower within 15 minutes on the very first day just from accelerometry data. In fact, we can predict before they even leave the nest what strategy they're going to employ. So lots of exciting further things to do. And with the advent of deep learning, we can also use drones because it's very expensive and difficult to catch animals. These are zebra. These are endangered. These are called Grevy's zebra. There's only a few hundred left. And so we're tracking them from drones. And we're using, this is not LEAP, but it's very similar to LEAP. It's a lot faster. It allows us to track the body posture of these animals from drones. We're actually also using deep learning to recognize who is whom, to do surveys of this population. Because normally you have to just go on the ground and look at the stripe patterns. But from the air, we could do surveys much more regularly. And of course, we can put the animals into the context. This is the real vegetation in which, the habitat in which these animals are living. We also want to track the predators. And so it's all very exciting. This is all happening in Konstanz, which is a beautiful old city on the border of Switzerland, which is why we still have such an established old city. They literally left the lights on during the war, so it never got bombed. It was believed to be in Switzerland. We have a beautiful lake overlooking the Alps. So it's about 15 minutes from Zurich Airport. We have a building that's going up now. This is a rendering. It's going to be constructed by 2021. This is, I think, the world's first building for studying collective behavior. And the idea here is we have computer scientists, biologists, psychologists, all working together in the same offices, in the same labs. And underground, actually extending well underground into the foreground here, we have a large animal facility, including an imaging hangar, which is 15 by 15 by 8 meters. We already have one that's 15 by 7 by 4 meters, where we can track at sub-millimeter resolution in 3D. Just got data yesterday from 20 birds, 20 starlings, within this large aviary, using Vicon and deep learning. We also just got a big grant, about 62 million euros. So we have lots of postdoc and PhD positions, group leader positions, tenure track positions, including neurobiology. If you know anyone that would be interested in, and it doesn't have to really be collective behavior because the brain is a collective, you know, it doesn't matter to us. Yeah, thanks to the people who support me.